Welcome to AMP2 Anatomy Labs. I'm going to just start off this week with a little bit of an intro to what you can expect with the Anatomy Labs. I'll talk about some of the systems. So we're going to go through 5 systems, just like we did in AMP1. The systems we have remaining are head. We're going to split head into 2 weeks. So it's going to be head and neck 1, head and neck 2. We'll do that next week. And then we'll get into the heart and the cardiovascular system in week 3, the abdomen and the GI system in week 4. And then finally, the urogenital system will combine the reproductive system and the urinary system for the 5th week of content. So nicely, most of the structures we'll encounter will kind of coincide to what we'll be doing in lecture, especially as we get to the kidneys and to the GI system. So that's going to be a nice correlation of structure and function throughout this course. Throughout this semester rather. So I'm going to start off today with lab 1, which is the first part of the head. We'll start by looking at the sensory structure, so the eyes, the ears, and the nose. We'll talk about some of the muscles of the face, the important ones. And then next week, we'll pick this up by talking about the thorax region, the neck region, as a precursor to moving into the chest and to talk about the heart in week 3. Now, the videos will be available on Tuesdays of each week, along with the quiz for that video. So you'll see both of those things available at the same time. Your duty is to look at the video, study it. I'm also going to include a virtual 3D video towards the end of the lecture videos, where I'll kind of give an overview of the important structures and really talk about the 3D sort of spatial relationship of all the structures we've encountered as a way to fit all of the structures into one piece and look at the bigger picture. So stay tuned towards the end of the videos for that 3D kind of overview. And then after you've studied the content for the week, you'll go and do the quiz for that corresponding week. The point breakdown is the exact same as AMP1. So for the labs, we've got 200 points for anatomy labs. We've got 90 points for the practical, which is a cumulative exam towards the end. And then we've got 18 points for each weekly quiz. And then we have our participation points that you will get for obviously participating, looking at the videos, and taking the quiz. That's, I think, 4 points each. That's a sum total of 200 points. The labs are supposed to be a way for you to study the structures independently this semester, given that we're online, but I will be available. I'm going to make myself available via Zoom. And I'll announce the dates of that. So stay tuned where I'll come on here on Zoom and I'll have like an open review session where you can come and ask questions and so on. So stay tuned for those hours as we go on. So enough rambling. I'm going to get right into lab 1, where we're starting here with the head. And the first structure we'll talk about is the eye. So the eye is obviously really important for us to take in visual information, sensory information about the world around us. And so the eye is set up and designed in that way, and we have 2 eyes, which makes us know how important vision is to our perception of the world around us. So some of the important structures that we can see right away when we look at someone's eyes are the pupil, for example. So the pupil is this black hole where light is actually able to come into the eye, and we can throw images onto the sensory layer that's called the retina, which I'll talk about in a minute. We can also see the iris right away looking into someone's eyes. This is the colored part of the eye. So if someone has green eyes or blue eyes, this is the area that is colored. We've got the sclera, which is the white of the eye. This is a fibrous connective tissue layer that surrounds the eye. This is what gives the eye its white appearance on the outer parts. We've got the cornea. So the cornea is the transparent region that is located anteriorly on the eyeball. So in terms of this sagittal view, this region here is the cornea. And the cornea, as we can see, is transparent, again, to let light in and help us with that sensory information projected onto the cornea. We've got the lens. Now the lens, and I think the tab for this one is off, but the lens is this part of the eye. This is what can control the location of where that light ends up on the retina. So the purpose of the lens is to focus the light exactly onto a specific spot on the retina. And in doing so, the lens can change its shape to be more convex or more concave in order to focus that light directly onto the focal spots here on the retina. Okay. I also want to point out the layers, so to speak, of the eye. So in terms of the sagittal view, we can really see the three important layers of the eye. So the outer fibrous layer is shown here in blue. This is the sclera that we talked about. The vascular layer, so this red layer is what is shown to represent the choroid. And the choroid is a vascular layer. This is rich with blood vessels, arteries and veins and nerves that are bringing oxygen and nutrients to nourish the eyeball. That's the choroid layer. And then finally, the innermost layer is called the retina, and this is the sensory layer. So this is picking up that information about images that are being projected from the outside world and turning that into sensory information, i.e. action potentials, and sending that information along by the optic nerve to the respective parts of the brain. Okay. Let's zone in here on some of the other important structures that are surrounding the lens. So we've got the ciliary body, which is all around the lens. And the ciliary body is really a modification of the choroid. So remember I talked about that vascular layer in red. That modifies itself as it gets in contact with the lens. And so this is a smooth muscle apparatus that can contract or relax, and that is what changes the shape of the lens and can focus light exactly where it needs to be on the retina. The connection of the ciliary body to the lens happens via the suspensory ligament. So these white ligamentous attachments here are called the suspensory ligaments, and those are what are actually anchoring onto the lens and moving with the smooth muscle adjustment of the ciliary body. We also want to point out two of the chambers associated with the eye. So we've got the anterior chamber, which is in front of the iris. So this is the iris here. And although it's being shown as a cut section here, remember this is a circular structure that's going right around the perimeters of the cornea. So anterior to the iris here, we've got the anterior chamber, and that has aqueous humor. So that's a kind of watery liquid humor or substance that helps to keep the eye open and nourish the structures anteriorly. Similarly, we have a posterior chamber, which also has aqueous humor, but that is going to be behind the iris. So we've got the anterior chamber in front of the iris, and then the very small posterior chamber, which is anterior to the lens, but posterior to the iris. Okay, and then the third chamber we've got is called the vitreous chamber, sometimes called the vitreous body. And this has a larger amount of fluid. This is more of a jelly-like fluid called vitreous humor. And the purpose of vitreous humor is to maintain the intraocular pressure, essentially preventing the eyeball from collapsing down on itself. So the vitreous humor is really important in keeping the shape of the eyeball, keeping it nice and open and patent with that more thick or more viscous jelly-like substance in here. Other structures here we want to point out the retina. So I talked about the retina earlier, but the retina is the sensory innermost layer of the eyeball responsible for picking up the light, picking up the sensory information that is projected from the light that is entering our eyes and making sense of that and projecting it via the optic nerve to the respective areas of the brain. We've also got the fovea. And the fovea is kind of this focal point where light projects to. It's a dip in the sensory layer here. It's going to be found lateral to the optic nerve. We'll talk about what the optic nerve opening, what this opening is called in a little bit. We've got the choroid, which I talked about before, that vascular layer that is nourishing the eye. And then we can also see the optic nerve here, which is obviously the cranial nerve 2, which is responsible for the sensory perception of our vision. So putting it all together, let's talk about how these structures fit together to allow us to process vision. So the focal point is where light needs to be projected. So all of the information that comes into our eyes as light is projected onto the exact region that's called the focal point, which is on the sensory layer of the retina. Now there are certain structures that help to focus this light. 70% of this focusing happens via the cornea, so the transparent layer here, but then 30% of that is done by the lens, which I talked about when it adjusts its shape. And we can speak about different pathologies that come about if light is not being projected onto the focal point. So if the lens or the cornea is not projecting light where it needs to be, we can be nearsighted, and that light can be focused proximal to the focal point, or we can be farsighted where that light is focused beyond the focal point. And that's what brings about a condition called astigmatism, where the cornea distorts the projection of light, and so it does not fall exactly onto the focal point. And that is obviously going to bring about blurring of the vision, either when we try to look at objects that are near, in the case of nearsightedness, or when we try to look at objects that are far away, they become blurry depending on where that light is being focused. We also want to talk about the optic disc. So I talked a little bit earlier, I alluded to a little bit earlier, the opening where the optic nerve enters or leaves the eye, that opening, let's jump back here. This opening is called the optic disc. And that corresponds to an area on in terms of our vision, called the blind spot. Now there are lots of cool experiments that you can use to actually find your blind spot using your finger or a piece of paper with a colored object on it usually. But your blind spot is the area where there's no visual perception in this spot. And that's because this is an opening where the nerve is actually coming into the eye or exiting the eye, depending on how you want to look at it. So this is called the blind spot, and it's created by the opening here. This is also appreciated as the optic disc.